So looking a little bit more at that skeletal muscle contraction, we were talking about how it grabs and pulls with the actin and the myosin, and how the troponin and the tropomyosin were assisting there. So we're going to get into a little more details here. The sites that myosin grab on the actin, they're not always exposed. It's not always ready. We don't want our muscles to always be contracting. Actin has to basically be primed for myosin to grab it. Contraction is when the myofibrils slide together, those Z lines come closer together, and remember they close up kind of like an accordion. We call that the sliding filament theory. Muscles need a stimulation. They need to be told to contract, and so this comes from the nervous system. And so contraction results from the movement within the myofibrils, actin and myosin, sliding past one another, okay, which shortens the sarcomeres and brings those Z lines closer together. This is a real important part of this unit here for lecture is understanding skeletal muscle contraction. And so I want you to take your time and really read through this. You may want to watch some extra videos. If you still feel lost on it, let me know, and I will find some other resources for you, or pull up another lecture. We'll get together and meet on it and talk about it, make sure we can kind of get a good understanding of this. So each neuron here has a process that's called the axon, and that's what we're looking here is the neuromuscular junction. This is going to extend from the cell body. So we have the axon like this, and then we have a cell body, and there are different types of neurons, so they're not all like this, but that's kind of a general structure. This here would be the axon, and this would be the cell body, okay? So it's able to conduct or send electrical impulses, and we call those an action potential. Each cell is called by what we call a motor neuron, and it has a functional connection, we would say, to the end of an axon, which means it has a connection, but it's not a physical attached connection. It actually has a gap there, and we call that gap the synapse. So it's not physically connected, but it's close enough to go ahead and transmit the message. So it's kind of like talking into a phone. So the space between the axon and the muscle fiber where they meet is called the neuromuscular junction, and that is what we're seeing in this picture here. So this would be the end of the axon, and this would be the muscle cells, and this space in the middle is the neuromuscular junction. We also sometimes call that neuromuscular junction the myoneural junction. Myo meaning muscle, neural meaning nervous system. So the space between the axon terminal and the muscle fiber where they meet, again, that's called the synaptic cleft. And it's just that opening, that blue space that we can see in our picture. An action potential sends a signal down to the axon, and it causes a neurotransmitter here. See if I can clean this up a little bit. It causes a neurotransmitter to be released. So here's our axon, and it releases a chemical. A neurotransmitter is a chemical here, okay? And so that chemical is going to then float, if you would think of that way, across that synaptic cleft and passes the message along to the skeletal muscle fiber. If you think about perhaps when you were a kid and you had the two cups with the string in the middle and you could talk to each other, and the message actually kind of transmitted along the string, it's kind of like that, or like old-fashioned telephones where we actually had wires that transmitted, except it's not a physical connection. But you're going to have that chemical that is going to be released into the synaptic cleft, and then it's going to be picked up on the other side. So the first event in muscle fiber contraction, and you need to know the steps of muscle fiber contraction, all right, that will be on the test, is that acetylcholine, and that is our neurotransmitter, and that is right here. Let me circle that instead of trying to write it. So that's acetylcholine. Sometimes it's abbreviated ACH. Okay, so acetylcholine is going to be released from the end of the motor neuron after that action potential is sent. So an action potential must be generated. It has to start. A stimulus going down the axon of the nerve to the synaptic knob is basically what that action potential is. And so it's got another stimulus that triggers that to happen. The action potential goes down the axon and gets to the synaptic knob, and it stimulates what we call vesicles, and those are the substances here that store these little ones here. Those actually store the neurotransmitter. And so the message is going to be sent down the axon to the end here, and it's going to be an electrical signal, and it's going to stimulate these to open up and be spilled into the synaptic cleft, release that neurotransmitter, which in this case is acetylcholine. There are other neurotransmitters. Okay, so if we're in a skeletal muscle, it'll be acetylcholine, and that's going to cross the synaptic cleft. It's almost like a little moat around a castle. Okay, and so it's going to cross that synaptic cleft, which we sometimes also call a gap junction, and then it's going to bind to a receptor on the other side. So if this is the muscle on the other side, you've got a little docking receptor that will receive that neurotransmitter. Okay, so it's going to bind to the receptor on the other side, and that is going to continue sending the signal. It's going to continue sending the stimulus that's telling the muscles to contract. So it's sending the message on saying, muscles, you need to contract. And then what happens is that causes sodium ion channels to open up. Okay, sodium ion channels are going to open up. So remember, when we're talking about neurons, we have resting potential, and we have sodium channels and potassium channels. And as that electrical stimulus goes, remember, we have more sodium on the outside and a little more potassium on the inside. And so when those channels or these gates open, sodium floods into the axon. The resting potential is generally -70. And so sodium flow ends and it changes that potential. It makes it positive instead of negative, or more positive, we should say. And so you have to get to a certain threshold amount to get enough sodium to reach, to start actually and trigger and send the action potential. And then it'll flow down and trigger, it'll flow down this way and trigger the muscle cell to start to contract. If you want to think, if you don't understand the threshold amount, we'll think about this. If I'm going to topple dominoes, I could just barely touch it and it won't do anything. But if I push it just a little bit stronger, whatever it takes for me to be able to push that domino over, that's the threshold amount. And so that's what we need to open up these sodium ion channels to get them to open and allow this to go through. Both acetylcholine and norepinephrine can affect that smooth muscle contraction. Okay, acetylcholine and norepinephrine. In order to stimulate muscle skeletal fibers, we've got to have that release of acetylcholine into the synaptic cleft. Okay, I left this here to remind you to be sure to watch the video on myasthenia gravis. This is a chronic autoimmune neuromuscular disease, and it causes weakness in skeletal muscles. These are responsible for breathing and moving different parts of the body, including the arms and the legs. The name myasthenia gravis is Latin and Greek in origin, and it means grave or serious muscle weakness. So be sure to watch that video. You really need to watch all these clinical applications in the animations because they're extremely helpful. It just doesn't work well to incorporate those into the lectures when I'm recording them. So I'm putting it on you to watch those separately. So once the action potential reaches that sarcoplasmic reticulum, this is where calcium is stored. When it reaches that sarcoplasmic reticulum, it releases calcium ions. The calcium ions are really important here because what they do is they uncover the troponin. And so there are two proteins that can cover actin. And then remember, that's troponin and tropomyosin. And so they have to go and uncover the troponin. All right. And so remember, this little section there is the troponin, and the longer curly section is the tropomyosin. Okay. So troponin holds the tropomyosin in place, and so those two are connected. It's kind of holding it in place, and if you look at that picture closely, you can kind of see you've got your three little things here. That's the troponin, and they're kind of attached to the tropomyosin. So they hold them in place, which is the binding site for calcium. Okay, calcium binds to troponin in order to change its shape. It bends it out of shape. When troponin is bent back or pulled tropomyosin out of the way, it pulls tropomyosin out of the way. And so actin now has those little handholds exposed for myosin to be able to grab the little handholds. Okay, and so like I said, you don't want your muscles to always be contracting. And so you have to have a way to stop it and start it. And so this is basically like taking the safety off of your gun. Okay. So actin has to have those handholds open in order to be able to grab them. Myosin is ready then, and it grabs it, and it does what we call power stroke. It grabs and pulls. It grabs and pulls. You've got to have energy for that, right? So you got to have ATP for myosin to work, and it's got to release the handhold so that it can then go and grab the myosin and cock that trigger again. Because remember, you got to cock the trigger. So if calcium is still present, the cycle will keep continuing, all right? So that's really important. So as long as calcium is there, as long as troponin has pulled the tropomyosin off, that handhold is there, and myosin is going to grab it and pull it as long as it has enough ATP, all right? So as long as you have calcium exposed, or calcium causing the exposing, and you have enough ATP, this is going to continue. So I want you to think for a moment, what would you need in order for this contraction cycle to stop, right? What would you need for this contraction cycle to stop? What would need to not happen or not be there? So here we're going to look at the whole cycle and see how do we stop this cycle from continuing. So here at the number 1, and you can see number 1 is, I'm trying to find a color that's not in there. How about green? Number 1 is right here, okay? So for this muscle contraction to start, remember, the nervous system has to send that action potential signal down and tell the neurotransmitter acetylcholine to be released, okay? So these are stored in these little vesicles right there. That's your acetylcholine. So they're going to go to the membrane there of that, at the end of the axon, release the acetylcholine into the synaptic cleft, which is that gap, that moat around your castle, okay? And then they're going to dock or bind to these little receptors here on the sarcolemma of the muscle, all right? So that is the first section. So the action potential has been generated and it travels down to the T-tubule. Calcium, and number 1 is important. Number 2 is important, but not quite as important as number 1. You need to know that step. Acetylcholine is released into the synaptic cleft and is picked up by the sarcolemma. It's going to travel down, and then the next one that's really important is that calcium ion release. Calcium is released from the sarcoplasmic reticulum because of the change in voltage. So here we have calcium being released, and so these do travels on down the T-tubule, but acetylcholine and calcium, those are two of the major players I really want you to know, okay? So calcium is released. Calcium does what? Remember, calcium binds to troponin. It binds to troponin, and it uncovers tropomyosin, okay? So it forms what it calls a cross bridge, okay? It forms a cross bridge. And so it uncovers tropomyosin, and so remember, then that handhold is exposed, okay? That handhold is exposed, so myosin then can grab and release, and you're seeing that here. And this, it kind of says cross bridges form between actin and myosin. What that means is it's doing this grabbing and pulling and grabbing and pulling and grabbing and pulling. And remember, as long as that calcium is there, it is going to continue, and as long as it has enough ATP. So in order to get this to stop, we need what we call an enzyme, right? Remember, enzymes end in ASE. Well, acetylcholinesterase, fortunately, is what removes the acetylcholine, okay? So that is the enzyme that removes the acetylcholine from the synaptic cleft. So it removes it there, and so the calcium will no longer be triggered to be sent out, and that will stop the handhold on the troponin. The tropomyosin will then again bind and cover the active site, and you will no longer have the muscle contraction. Now, how else could you stop the muscle contraction? One, you have to remove calcium, right? Who did we say removes calcium? Right, it's this acetylcholine esterase. That's who removes the calcium. But there was one other thing we said you had to have for this to continue. What was that? The ATP. So if you ran out of ATP, this would also stop, okay? I know this section here that we're talking about is kind of abstract and it's kind of hard to focus in and understand these pictures, but you really need to pay attention and make yourself focus really hard on these, okay? Because you need to know this information. It's important. When these sarcomeres shorten, the thick and thin ones, again, remember, they slide past each other. And this is kind of a summary here. You can see you've got your H zones and your I zones, I bands narrowing, and then the Z lines move close together. And that's what this is showing you, okay? So this is the diagram here on this side, and this is what it actually looks like microscopically. So you can kind of get a view of what's happening there. I've kind of already explained this one. The myosin head is going to attach and grab that binding site, and that's what forms what we call the cross bridge when it's actually grabbing. ADP, remember, adenosine diphosphate, and phosphate are then released by myosin. So the new ATP can then bind to myosin. So it's kind of getting recharged there. And then that linkage between your actin and myosin cross bridge will break, the ATP splits, and that cross bridge can eventually go back to the original position. This basically already kind of, again, summarizes what we've already talked about, that acetylcholine esterate, decomposing and removing that acetylcholine, which stops the calcium from being sent down, which will then stop the whole cycle from occurring. So let's review all the main players here. And I'm talking about this one a lot because, like I said, it's kind of abstract and it's not something that we're real familiar with. So muscle fiber contraction. First thing that happens is excitation. That is where that action potential is triggered. Causes the acetylcholine, which is the neurotransmitter, causes the acetylcholine here to be released into that synaptic cleft at the end of the neuron. That's the synaptic knob is what we call that. So it's released and then it binds to the receptor here on the other side, right, which cause those sodium ion channels to open, and then calcium is released, which will go and uncover the troponin, which will then cause the tropomyosin to uncover, and then we're going to have the power stroke, okay? So basically we've got troponin and tropomyosin that cover actin or uncover it, calcium, which causes them to uncover it, actin and myosin that form those cross bridges, okay, and form that power stroke, and then ATP, which is going to come in and it actually helps cock that trigger. It takes energy to cock a trigger on a gun, right? So it takes energy to set that back so that it can go and pull again. It's kind of like setting a little mousetrap. You're storing energy there. And so for muscle relaxation to occur, we need that acetylcholine esterase, right? And that's going to remove the acetylcholine and stop that whole cascade of events, all right? So I hope I'm not repeating this too much, but it's really important. This is a really good illustration here also. So we've got two energy sources here for contraction, for muscle contraction, creatine phosphate and cellular respiration. ATP is needed for muscle fiber contraction, and if there's a limited supply of ATP, we must regenerate it, right? So the initial source of ATP, or initial source of energy, sorry, to make ATP from ADP and phosphate is creatine phosphate. This is a high energy molecule. It has a high energy phosphate bond, and so it stores energy in that high energy bond when it comes from the mitochondria. ATP then is broken down into ADP. Creatine phosphate puts the phosphates back on it, which makes it become ATP. So we constantly recycle and regenerate that. And remember, we call that phase or that transaction phosphorylation, when we're putting the phosphate back on. So the amount of ATP and creatine phosphate in a muscle is limited to enough for about 10 seconds. So we have to continually make more. So how do we make ATP? You should know that. Cellular respiration, right? We use glucose to make ATP. So glucose is stored in the cell as glycogen, and so we can break down that glycogen and convert it back to glucose. So that's really important that we're able to do that. So remember, cellular respiration, part of that we do need oxygen. How do these tie together? Remember, glycolysis is the first step of cellular respiration. And where does it occur? In the cytosol or cytoplasm, same difference. And it is anaerobic. So the first part, we don't need oxygen. Remember, it only makes 2 ATP per glucose here, okay? So only a little bit of ATP is made there, all right? So to completely break it down, we need oxygen. We've got to have oxygen for the next 2 aerobic steps, which are going to be the citric acid cycle and, of course, the electron transport chain. Blood carries oxygen from the lungs to the body cells, and then it's going to be transported by hemoglobin and released where it's needed. And so then it will be available for those aerobic portions of cellular respiration. Myoglobin is made in muscle cells and can bind oxygen. And so it has more of an affinity or an attraction for that oxygen than hemoglobin. So it's going to grab it from and steal it from the hemoglobin and store it for some later use for muscle contraction. When we're using skeletal muscles for a long time, oxygen can't be supplied in order to sustain that aerobic respiration. We have a limited amount. Metabolism then must switch over to the anaerobic production. This is what we call lactic acid metabolism. Lactic acid is produced and it will accumulate in the cells. And this is kind of what we associate with muscle burn. And we do know now that it is a little more complex than that, but it is associated. Lactic acid is going to diffuse out of those muscle cell fibers and go to the bloodstream in the liver, and then be converted to help to convert glucose. And this actually generates what we call an oxygen debt that needs to be repaid later, okay, in order to restore the balance. Oxygen debt is equal to the amount of oxygen that the liver cells need to use up that accumulated lactic acid with making glucose, plus the amount that muscle cells need to make ATP using that creatine phosphate, all right? So hopefully we're getting kind of to a more relatable portion here when we're talking about muscle fatigue and lactic acid burn. This should be some things you're kind of familiar with, okay? So if a muscle is exercised for a really long period of time without resting, it gets to where it can no longer contract. And this is where if you've ever run, when I was younger, I used to run, and every Sunday I would do a long, slow 10-mile run. And when you or a jog, I should say, when you get to a certain point, it's like your legs feel like jelly. They just feel like jello. There's just nothing there. That is muscle fatigue. This is caused by a decrease in blood flow, an imbalance of ions that are going across the sarcolemma. Okay, and that's from the transfer of ions during repeated stimulation over and over. And more importantly, from accumulation, I should say, of those hydrogen ions because of that lactic acid. It's an acid, right? So remember, acids release hydrogen ions. Okay, so it's got accumulated hydrogen ions, and that is going to change the pH. And so that prevents, that change in pH prevents muscle fibers from responding to stimulation. Because remember, that starts with an electrical current, and hydrogen ions have a positive charge. So lactic acid can build up and cause muscle fatigue. How quickly that happens depends on the type of muscle. We have fast twitch fibers and slow twitch fibers. Sometimes muscle fatigue and cramps can actually happen at the same time. What would cause a muscle to stay in contraction, right? If the acetylcholine wasn't removed, perhaps, right? There are different things. Cramps are uncontrolled stimulation. Are some ions not being removed? Are too many others put in? So there are several answers here, and we don't really quite fully understand all of them. So remember, heat is a byproduct of cellular respiration. All active cells generate heat. Muscle tissue plays a large role in supplying body heat. A little less than half the energy released from cellular respiration is available to be used for metabolic processes. The rest has to be released as heat. Blood transports heat from muscle contraction throughout the body, helping maintain that body temperature. The more active you are, the more heat is released. Remember, to cause a muscle contraction, you've got to send a stimulus that reaches a certain threshold. And we can have things like these TENS units that you can attach. I actually have one sitting right here. You can attach something like this to your muscles and turn it on and facilitate a contraction that way as well. So you got to send a stimulus, but it's got to be of the right threshold amount, right? Remember, I was talking about being able to topple a domino, which is a very small example. Let's say there is a piece of furniture you want to move. If you just barely push on it, you can't move it. There's a certain amount of strength, a threshold amount of force that you have to push in order to get that moving. All right, that's what a threshold is. How much do you need to get this started? So it's got to be high enough to trigger a response and generate that action potential. Once that action potential is reached, the action potential is generated throughout the muscle fiber, causing enough calcium ions to go into the sarcoplasmic reticulum and contract the fiber. A single action potential going down a motor neuron normally releases enough acetylcholine to bring the muscle fibers to that threshold and generate an impulse in the muscle fiber. So this contraction of a muscle fiber, this is what we mean when we say a twitch, a muscle twitch. We said we had slow twitch versus fast twitch. Okay, so it's how long is this going to take to happen? So this includes contraction, where the fiber pulls at the attachments, and the subsequent relaxation, which is where the pulling force starts to decline. We use a myogram to record this, and there's 3 phases of a muscle twitch. We have the latent period. This is a delay time between the stimulus when it's applied and when the muscle responds. Okay, that's the latent period, and you can see that here in blue. Latent period, you're sending it, but nothing has happened yet. All these events are taking a little bit of time to occur. This is just under 2 milliseconds, so just a very, very small period of time for a human muscle. Then we have the contraction period, where it's actually contraction, and then the relaxation period, where it begins to relax. We also have a refractory period, and that is a time when the cell cannot repeat the action. You can't send another signal to contract, okay, which in this case is muscle stimulation, is that muscle contraction. A muscle fiber brought to threshold is going to have an all or none response. If you put a less amount of force or a lesser signal, it's not going to contract on a smaller amount. You either start it or you don't start it. There's no in between, and that's what we mean by all or nothing response. You send a signal that's strong enough to turn it on, or you don't. Okay, so it's like you can, most lights, you can flip the switch and turn it on, or you can turn it off. This would not be like something that has a dimmer switch. Okay, this is turn it on or turn it off, no in between. So for a sustained contraction, the force of those individual twitches can actually combine into what we call summation. We can sum or add them together. Okay, if the contraction is forceful and sustained and does not allow any relaxation, we call this tetanic contraction. And this is what happens when you have tetanus. Tetanus is lockjaw. You have a long sustained contraction and there's no relaxation. We also have what we call recruitment of motor units. A motor unit is a motor neuron and the muscle fibers that it controls. So it's those put together. The number of muscle fibers that you have in a motor unit vary. The fewer the muscle fibers that we have in the unit, the more precise the movement can be. Okay, so single motor neuron here can have as few as 4 fibers, or it can have thousands of muscle fibers. Some of these muscle fibers are more easily brought to a threshold than others. They have a lower threshold. So a muscle fiber that is exposed to a series of stimuli that keeps increasing frequency and is summed, that combines those individual twitches, then it can result in that complete sustained contraction. If more easily stimulated motor neurons involved, we only need a few motor units to contract. With higher stimulation, other motor neurons respond and more motor units are activated. This increase in the number of motor units is called multiple motor unit summation or recruitment. And so that signal basically has got to go somewhere. And so if they have a low threshold limit, they'll go to other motor units. With a high intensity muscle stimulation, recruitment can continue until all the motor units in the muscle are activated. That's how you would activate the entire muscle. So if we have a smaller motor unit, and that means a smaller diameter, these are going to be easier, the ones that are easier to stimulate, and they're going to be recruited sooner. A larger unit is going to have a larger diameter, and it will respond later, but it will also, because it is larger, it's going to have more force. So summation and recruitment together can give a sustained contraction of increasing strength. Individual twitches don't usually occur. Several muscle fibers are usually going to be responding together to make a smooth movement instead of a jerky contraction. Even if a muscle seems to be resting, it has a small degree of sustained contraction, and this is what we call the muscle tone. Now, this is not the same as what we use in our everyday vernacular for muscle tone, meaning somebody's in really good shape. This is partially how we maintain our upright posture. Those muscles have to be partially contracted. The neck, trunk, and lower limb muscles must remain taut, in other words, in order to hold your body upright. Muscles are going to either shorten when they contract, or they can lengthen, or they can stay the same in what we call an isometric contraction. And so isometric is when the muscle contracts but does not change length. We also have isotonic, where it contracts and does change length. Eccentric occurs when the force of the muscle is less than what is needed to move or lift the object, like laying a book on the table, and that's going to be a lengthening contraction. It's not enough force to shorten the muscle, so it lengthens. Sometimes muscles contract and the parts it is attached to do not change length, like pushing it against a wall. Muscle tension increases, but the wall doesn't move, and so muscles remain the same length. Okay, that's isometric. And then we have, which one have I not talked about here? The concentric. I don't think I talked about that one. If I did, I apologize. I'm repeating it. Concentric is where you are going to have the muscle contract with more force. You're really lifting something, and so that's going to cause those muscles to really shorten. I know this is a longer lecture for online, but we're almost done. Just hang on, pay attention, pause to take a break if you need to, okay? So we have talked a little bit about fast twitch and slow twitch muscle fibers. Let's look at the difference between those, all right? Muscle contract at different speeds, and whether or not they make ATP by, well, oxidatively or glycolytically. So how do they get their ATP? So we have three basic types, three combinations here. We have the slow twitch, which is going to be red muscle fibers. This is found in long muscles of the back, for example. Red fibers have a red pigmentation from the myoglobin. They have lots of oxygen-rich blood, lots of mitochondria, and they can contract for a long period of time without fatiguing. Fast twitch, which are glycolytic, are more white fibers, so that's type 2A. These have less myoglobin and a poor blood supply. So these are found in areas like hand muscles and the eye. They have less mitochondria, more sarcoplasmic reticulum to store calcium, and their ATPase works faster. They contract rapidly by fatiguing when lactic acid builds up, or they contract rapidly and fatigue when lactic acid, tongue-tied, I'm trying to be quick for y'all, lactic acid builds up. And then we have fast twitch oxidative. These are red. These are intermediate fibers, so they're kind of in between. They have the fast twitched speed, but they need or they have more oxygen capacity, so they can store oxygen more. Activities like swimming a long distance or running long distances are more likely to stimulate the development of slow fatigue resistance fibers. Birds that migrate long distances have abundant slow twitch fibers. This is why their flesh is so dark. This is where dark meat and white meat comes in. So chickens, though, see, they sit on the ground and they only kind of flap around the barnyard. They have a lot more white meat because they have fast twitch muscle fibers. So a world-class distance runner is the human equivalent of that migrating bird. Their muscles have more than 90% slow twitch fibers. So using your muscles is important. Skeletal muscles respond to use and disuse. Forcefully exerted muscles, what do? Why do people work out? Well, to build muscle. And we call that hypertrophy. Remember, hyper means above. Unused muscles, though, experience what we call atrophy. And you've seen this probably if someone broke their arm and it was in a cast for very long, and it's much, much smaller because it has atrophied. It has decreased in size. It's decreased in strength. When a muscle undergoes atrophy in response to disuse, there's also a reduction in capillary networks. The size of actin and myosin filaments decrease and the muscle just gets smaller. So that's the last part of this section.